here, and I would like to welcome you all to our first workshop on application of quantum mechanical and molecular mechanical simulations and using this in NAMD. This is actually a new feature that was added to NAMD because of the huge interest and the large number of requests that we had from the community. We want to do QMMM, we want to do some chemistry inside the system, and then we implemented that, and now this is the first workshop we have for teaching people how to use it. So before we go there, as you can see from the program, we're going to have some introduction on the MD part, which is the hardcore of NAMD. You do MD simulations. If you just want to do quantum chemistry without bothering with the environment, you won't be here. So there are much better programs and algorithms and workshops to go to. So you're here because you're interested in doing chemistry inside large biomolecular systems. Therefore, we need to make sure that you know the MD part and how to deal with the molecular system first. That's why we decided to have the first day dedicated to really being able to work with NAMD, why we do MD simulations, motivate that, and have some examples. And then we can move to the QMMM. So before I go on, I would like to actually just show a few slides. We are in our center area, and I think I would like to highlight some of the important aspects of our operation. So we are a center, for NIH Center. We are here to serve the community and help the community because there is an increasing need in using molecular dynamics as actually a very sort of prominent method in modern structural biology. So if you see, it's not just computing people, many structural people, experimental scientists would like to be able to use MD in their research. So this is the number of papers, and you can see it's almost exponentially increasing. So MD is being used a lot, and we would like to teach people and provide the technology for people to do it better, to do it faster, more efficiently, more accurately. So these are the two programs of the center, VMD, the visualization program that you will use a lot. Not only us, almost all MD practitioners use VMD because of its strength in handling large data sets and a variety of analysis and everything. You can see we have more than 100,000 users by very conservative measures. And 19,000 users of our MD program. Many of them are NIH funded, and people are actually using the material on our websites, including the tutorial examples and everything. If you don't trust me with these numbers, you can look at the number of publications that cite our code. This was updated, actually. I don't have the update slide. Didn't get a chance to put it in. But actually, if you look at this, you can see that people are using our software. Many of them use them and don't cite or don't publish. Those that published, some of them don't cite even, but even if you just look at people who use the software and publish paper and cite us, because the journal allows them to have more references, no limitation, you can actually translate this number. Every 2 hours, we get one citation. The actual number is now 1.5 hours based on the updated information. But so that by the time we are done with this workshop, I think we should expect another, about 50 other citations. So people really use the software, and we are very proud of that. So we try to make the software available on all computational platform, your laptop, every new supercomputer, platform that comes online, even if it's before it is available, we try to tune the software there so it runs perfectly there. We are constantly adding features, excuse me, that are requested by the users. This is a great example. QMMM was something that people asked, and we are adding it. We cover a variety of systems going all the way from molecular orbitals, quantum mechanical results, all the way to cells. And then we are spending a lot of time, and you see some examples today, to make the software easy for people to use. Maybe you don't want to invest, I don't know, a month to be able to put together your simulation system. We have the quick MD that quickly helps you to put it together. Maybe you don't want to even set up your hardware, so we allow you to run on the cloud. This is something we're going to practice also for this workshop. So that way you don't even have to set up a machine and configure it and worry about it. That way you take the examples that you start working here. When you are in your lab, you log into your Amazon, you get the same instance, and you can continue working on them. So we try to actually make it remove all these barriers so you can actually quickly start using the method and making scientific discovery. That's the idea. We invest a lot of time actually being aware and being on top of the hardware technology in the field, working directly with NVIDIA on their sort of next generation hardware. All the supercomputing centers are directly working with us, and we are involved in the sort of early implementation or installation of their hardware and testing our software, because our programs are actually really top programs that are used by the community. And a variety of other technologies, Amazon Cloud, virtual reality, remote visualization. These are all the technologies that. So I mentioned actually we try to make our software easy to use. This is already, I mean, you will see actually a long lecture on this QuickMD and how it allows you to set up your simulation and your QMMM calculations and visualize and analyze the results. But this is another example in which we are trying to make it very easy for experimentalists who want to fit a molecular structure into an electron density coming from cryo-EM, for example. You can see that we can interactively start your simulation from this interface, and then using a method we call MDFF, I will talk about this a little bit. You can actually apply force. You can see this arrow here that the user grabs part of the helix and just pulls it into the density. That way you have a much more interactive way of fitting your structure. At the same time, you can monitor the cross-correlation between the structure and density. And this is something an experimentalist who doesn't want to spend a lot of time to learn all these details and the scripts and the files, they can actually use very, very easily. That's the idea. So we also teach people, train people. This is one example here. We are very proud that we have given 50 workshops already. So this number is really impressive. I mean, you don't know how much work goes into setting up and giving workshops. So usually centers have, let's say, 1, maybe 2 a year. We usually have 3 to 5 workshops a year. And we have trained a lot of researchers, students, postdocs, faculty members, people from industry who want to use, and a lot of material actually on the web that people are using. But okay, so that was a very brief introduction of the center. Let me now go to the subject that I'm supposed to cover this morning, molecular dynamics. As I said, I definitely firmly believe that this is a key methodology in modern structural biology. So if you really want to understand molecular systems, the first step is to get their structure, but then that's not enough. You need to know how things are moving, because if you freeze organisms, they die, they don't work. So we need to take into account thermal fluctuation to understand the function of biomolecular systems. And NAMD has been extremely efficient because it gives you both high spatial and temporal resolutions. So that's really something that no experiment can match. In principle, no experiment will be able to match this because of the uncertainty. But actually, computationally, you can go to any temporal resolution you want. And then spatially also, we have a very, very high resolution. You can keep track of individual atoms every 1 femtosecond. So that's really amazing. Of course, it is a model that we try to make it as accurate as we can, but still it's a model based on the input. So it's not perfect. So there are some approximations that go into this business. One of them is the lack of electronic behavior for most part of the system. So we don't have polarization, we don't have proton transfer, and that's something we try to address by including quantum mechanics, embed a quantum mechanical part into these molecular systems. But for the most part, we don't have a very sort of high accuracy potential energy or Hamiltonian going into this kind of calculation. So that's one area. The other area is that because they are so expensive, we are limited in the time scale. So that's something, again, these are the two most important aspects you should keep in mind. Otherwise, it's really powerful, especially if you couple it to experimental results and work together, test the results through various sort of ways. And I'm going to show you a couple of examples how we did this for some of the systems, and that became really made the results much more meaningful and much more appealing to people. So I'm showing actually a couple of examples here, and I'm going to cover actually how we have used MD, for example, for this example. This is my most famous movie. I made this in a cold room. It took me 3 days to make that. Nowadays, you can make something like this in 10 minutes on your laptop. So I had to go sit next to a workstation. It made it to the Nobel Prize website. So this is the closest I get to the Nobel Prize. So, but that here actually we are showing an equilibrium simulation in which you can see water molecules without any force applied, any tricks, simple thermal fluctuation. You can see how water molecules going through a membrane channel. The membrane is sitting here. And then you can see it's a random walk that we have read in textbooks. These are sort of, they go up and down without realizing what they are doing. And every now and then it happens that all these 10 or 12 water molecules move in the same direction. And then that translates in the net translocation of one water from one side to the other. So that's how it happens. A lot of failing, trying, until something sort of translates in meaningful effect. So you can see this. This is the only method that allows you to watch this in real time. There is no other method that allows it. So equilibrium simulation, if the time scale of the phenomenon that you're interested in is short enough, then you can nicely capture it with MD simulations of several tens or hundreds of nanoseconds. This is 5 nanoseconds. In fact, what you're looking at is 0.3 nanosecond segment of a 5 nanosecond simulation. Sometimes things are slow, so this is the example of a large protein conformational change in a drug exporter. So this guy binds a drug that is inside the cell here, and then the drug binds here, and then ATP binds here, closes this part, opens the other side, and ejects the drug to the other side. It exports drugs. P-glycoprotein, a major problem in cancer chemotherapy because it pumps anti-cancer drugs out of the cell. And people want to understand how it works, finding out all these intermediates that are involved in the function to be able to maybe design an inhibitor to block that to make anti-cancer drugs more effective. So we want to understand conformational change. This is just an example of slow conformational changes that you can't capture by doing simple, microsecond simulations. Now, this is usually millisecond or longer. So in those cases, we have to do other tricks. We have to do what we call non-equilibrium approaches. We have to bias the system in a very careful manner, of course. But then if you do it carefully, then you have the chance to maybe go and explore other parts of the configuration space and get some idea and information about how this might take place. Here's another example of where we use actually MD not just to simulate the whole thing, but just to construct a model of, let's say, cell, for example. It's sort of very prototype kind of model of the entire cell. Envelope, I should say, there is nothing in it, just water right now. So you can see a lot of lipids, a lot of proteins. Just building this model, if you don't do it carefully, it's going to collapse and not stable. In order to do that carefully, you have to do a lot of refinement of lipids and proteins when you place them, when you want to actually carve a position or a hole for proteins to place them, how we pack lipids, how we come up with a relaxed structure. We use molecular dynamics a lot for all these steps because that allows us to have a much more relaxed and sort of structure that has the right chemistry in terms of side chains and everything. So there are some examples of how you would use MD. So then I think the question is, so why you are interested? So sorry, I forgot to remove this. Computational structural biology, what we call, so essentially describing biomolecules at the nanoscale. So why are you even interested in describing a molecular system at the nanoscale? So why do you need to know these things? Here is one example. So we want to understand actually mechanisms in molecular biology, molecular basis of disease. Drug design is a very important aspect, and of course nanotechnology, and whether we can learn something from these molecular systems and apply them to nanotechnology is another application. So I remember I was at an NSF meeting, and somebody had made a very interesting movie about the nanotechnology and about some nanomaterial. And it was really enjoyable, and we watched it, and then people asking, why don't you make more of these movies? So impressive. And he said, the problem with nanotechnology is that you can't see it. It's so small, you cannot see it. The movie he made was mostly based on computational models. Based on information, you put them together in a computational model that is reflecting what is happening in reality. So there is no way you cannot watch how things go through a nanotube by microscopy. You might be able to freeze it and take a picture, but that doesn't tell you the whole story. So really nanotechnology, doing it in a sort of effective way and in a rational way, heavily relies on computational models. The same applies, what we are looking at, molecular systems, molecular biology, in a way is a nanotechnology kind of application. Here is an example. So this is a drug, actually a drug that is used in the market as an antidepressant, and you can see where it binds exactly to its receptor. By blocking this transporter, this drug actually increases the concentration of dopamine or serotonin in your brain, and that removes depression. So now we know exactly where it binds, what are the amino acids that are interacting with this drug, and what happens if a mutation happens here, how we might be able maybe to improve the binding affinity of this drug by changing the chemistry here. So this is great. So this is something we would like to understand biology at nanoscale that allows us to approach problems of drug design and diseases much more rationally. So here is another example. This is a paper we published actually several years ago. Back then, it was really the first example of a real ligand, in this case a nucleotide, binding to its binding site using unbiased simulation. And you can see actually how much changes you can see. Initially, you can see all these residues are doing their own things, but then once the nucleotide comes in, the phosphate groups here so they start to interact with these basic side chains. You notice actually the base part tried to dock in, but it couldn't complete. Yeah, you can see now these guys are approaching the second phosphate groups, and now this docks in nicely, and you come up with a stable configuration. So really the binding pose of drugs, so either you have to crystallize this under crystal conditions, or you can actually do MD simulation. This is something that has become more and more, we have more examples of these days. So you really, the most detailed microscopic view. And then we explain, of course, in addition to just see how it happens, the structure, you can get a lot of additional information. This is the electrostatic potential map of this area, and you can see here where you attract the nucleotide coming in, there is a huge positive potential here caused by these basic side chains actually that pulls the nucleotide, and that's why we could see it in unbiased simulations. And then actually we showed that this is the case for the entire family of these transporters. And very interestingly, all their substrates, nucleotides, oxalates, all the phosphorylated groups, they are all negatively charged in mitochondria. So that was really made a nice discovery about the whole class of proteins. So really, this is yet another example. So now, more recently, so this is a animation showing a GPCR, G-protein coupled receptor, a major site for many drugs that are in the market, probably one of the most important classes. And this is a simulation done by the group of the Shah. So they have the specialized machine, Anton. They can easily go through many, many microseconds of simulation. So you can see the time scale here. This is animation that they have as a supplementary information there. And in this case, we don't have a strong electrostatic pull like what we did earlier. This is a sort of ligand, so you're just relying on simple diffusion of the ligand in solution. So you can see actually that and you can watch how long this, you see it's sort of initial binding, it's not quite there, so it takes more than a microsecond to find. You notice actually, of course, it happened very fast. So it took about 5 microseconds or a few microseconds to find the right binding. This was done before this structure was crystallized with the ligand. And once the crystal came out, they're right on target. So it's really impressive. Yeah.
Just a quick question. In the previous video and in this video, were there several ligands in the solution then? Like, did they set it to a certain concentration of ligands?
So in the previous one, I think we had 2 or 3 only. So because this is heavily charged. In this example, I'm not sure, but I think they only had 1. And that you are asking, actually, you're pointing out a very important point. So in reality, you usually have many, many copies of the ligand, so they come and try and go. But in this for a very small simulation, unless we go to unrealistically high concentration, so you have a very small water box here, even 1 ligand in there is a very high concentration. You can do those things. We use those tricks. You should do those. But that would be for people who are too concerned about experimental conditions. They ask you, oh, your system has, I don't know, 2 molar salt. Why did you do that? Is it going to destroy the system or not? No, in this case, it was only 1. So now, again, I already pointed out about this. I think I'm not going to explain this again. We would like to be able to capture intermediates. So from crystallography, we only see maybe 2 structures. If we can describe how this thing moves, we have a much better chance to design a specific drug for this compound. I mean, the main problem in drug design and drug discovery is not finding something that binds protein A. It's finding something that doesn't bind proteins B, C, D, E, and even 500 other proteins. Really, specificity is very important. So you can always find something that interferes with this. But if that thing also binds other proteins, you will have side effects and cannot use that as a drug. So therefore, you have to search for a lot of options to hopefully find something that only binds this with high affinity and nothing else. If you have 1 structure, your chance is limited. If you have like 20 different intermediates, you have a much higher chance to find something that specifically interacts with this machinery and not others. So that's the idea of what people are doing. Instead of docking into 1 conformation that you get from the crystal structure, we would like to describe the dynamics, hopefully the process of conformational change over the functional cycle, and that gives you a lot of material to work with for your docking approach or whatever you want. So this is yet another example that we sort of use the non-equilibrium systems, in which we are using actually these 2 helices. You can manipulate the system extremely well using a variety of what we call collective variables, sort of system-specific variables that allow you to induce kind of different flavors of conformational changes in bias simulations. And then watch, for example, how water content of this half changes and how water content of this half changes as you go from inward-facing to outward-facing form of this protein during the cycle. And you can see, I can monitor exactly how many water molecules are here, how many are here, and show it nicely goes from one state to the other. In fact, it has to close on one side before it opens to the other side to preserve the concentration gradient. That's a very important aspect. It's called alternating access mechanism. You should alternate. You can't be open to both sides, otherwise it becomes a channel, and the gradient is going to dissipate. So this you can monitor. So again, I repeat, no experiment can give you this detail. Watching how many water molecules are in here and in here. There is no experiment in that. Oh, that's okay. Okay, I thought you had somebody had a question. So in terms of nanotechnology, I already made the point. So this is how nanotechnology is done now. Oh, I want to make a device coated with antibodies to be able to detect viruses, for example. So this is how my colleagues are working. This is the figure they put in their papers. Hand-drawn. Are you sure you have this? Are they all standing? This is, and how can you improve this design by this Mickey Mouse cartoon thing? That's why. The problem is it's so complicated, but we want to move in that direction. So that's what you see there is actually we haven't done much simulation. So these antibodies are connected by polyethylene glycol, sort of short polymers to the surface. And then you have, see, this is the first model we tried to put together. I'm going to just go through this animation. You can see these PEG molecules, sometimes they are connected to antibodies. Some of them that are not connected collapse and cover the surface. Hydration pattern is going to be different. And then all these antibodies can have different conformations. Sometimes they come too close to each other and start interacting with each other. I can't tell you that we have discovered anything useful yet, but at least now I have a realistic model for, for this. And then again, like this, you can start asking some questions. What happens if you change the concentration? What would be the optimal density? How about the length of these things? What happens maybe if you can longer and shorter? And that kind of question. You can start asking meaningful questions. Here's another example. Gold nanoparticles are used a lot as a kind of substrate for absorbing a lot of material for drug delivery. And you can see this is an experimental result obtained by my experimental colleague, Kathy Murphy here in the chemistry department. And this is what they get. And then they put this, again, a hand-drawn or power pump generated kind of a schematic there. So we would like to move away from here and be able to use modeling and simulation, MD simulation, to create a reasonable, also more realistic representation. So now you can see gold atoms. So now I'm going to cover them. We have covered them by sulfur atoms that are used to connect the, again, polyethylene glycol. So now, and then you can actually put it in a water and you start simulating them. Maybe we can, you see the water hydration pattern on the surface, shown in the surface representation of how these things move. And then on top of that, I can add the lipids using, again, some methodology that, some tricks that we know to sort of allow them to aggregate in a natural way on the surface. So these are lipids initially scattered in the system, and now you can see we are using a sort of external grid potential, very powerful way of inducing non-specifically kind of motion of atoms, and you can see this is how this looks much closer to what this thing looks like in reality compared to this that has no predictive power, essentially. And here is an, yeah, so these are some other kind of phenomena. So you can see how proteins might bind to the surface of a lipid bilayer, what part of the protein is interacting with lipids, why those lipids are important for the function of the protein. This is hydration pattern at the interface between some viral shell proteins that might tell you about, I don't know, drug penetration. And this one is just a thermal fluctuation of the lipid bilayer. So again, so I think I'm just give you some examples of what kind of information you can expect from these simulations. Excuse me. So this is a more recent example we did together with a group of Eric Gore about a year ago, more than a year ago now. So they have the protein. It's an open state of an ion channel. But if you just look at the protein sitting in the membrane now in this simulation, if you look at the protein, it's completely closed at the bottom. It doesn't look like an open channel. And then you have these big holes or openings or windows or fenestrations on the side. And the question was, how does this protein work? I mean, they did a great job. He's a very, very good crystallographer and a structural biologist. He has obtained the structure of many, many membrane proteins, key membrane proteins. And now they got this new state, and then, of course, I mean, the question is, how can this be an open ion channel? So we also looked at the structure. It's not clear. Let's put it inside the membrane and simulate that. And then we can put an ion. We have physiological concentration of ions, apply electric field, mimicking membrane potential, and watch where the ion goes. And as you see, actually, the ion, it's interesting that lipids have a significant perturbation. These are the head group of lipids. You can see they come all the way to the midpoint of the membrane, almost to the other side, lining those fenestrations. I'm not showing the lipids in the front that do the same. To this front fenestration, there are two other fenestrations, one to the right, one to the left, one in the back. And they see how they line and complete the permeation pathway. I think this is the first example in which we are showing, MD is showing how lipids directly participate in the function of the membrane channel. So it comes, the ions again, is handed between here, you see, interacting with that head group, this head group here. So these lipids are directly participating in the function of the protein. And then it comes out kind of through this fenestration. So, yeah, I think I talked about this already, but let me actually emphasize major limitations. Remember, time scale. We are limited by the time step that we can take because you want to describe the fastest degrees of freedom in your molecular system, which is the bonds between hydrogens and heavy atoms. You have a low sort of small mass on hydrogen. They move very fast. You have to take very small time steps when you are injecting thermal energy into the system. Otherwise, they're going to move like crazy, and the simulation is going to crash. So we have to take 10 to the minus 15 second time steps, which means every 10 to the minus 15 second, you have to calculate all the forces and everything, and that makes it extremely slow. So we are limited in terms of time scale. That translates into limited sampling. That's one big problem. I guess if somebody can solve this problem, we can expect the next Nobel Prize because that's going to tremendously expand the scope of what MD can do. And the other aspect is force field approximation. Most of the things we do are classical. And of course, there are problems describing a complicated electronic structure with a handful of numbers, fixed charge, and everything. So these are the two major issues. People are working on this. Of course, we want to make faster and faster algorithms that can take advantage of supercomputers and in a better way. And we have always improved versions of force field, better and better force fields. We have polarizable force field now. And again, but then sometimes say, you know what, I have to go back to much higher levels of theory, quantum mechanical descriptions for my problem. And that's the whole idea of QMM. Okay, so now let's say if you want to set up an MD simulation, so these are kind of major steps. Are they doing NAMD tutorial this afternoon? Yeah, okay. So these are the steps, and I think you're going to walk through these steps for a small protein this afternoon just to learn how to work with them. So you just have to prepare the molecular system. That means you get your PDB from the data bank, and then you have to generate a PSF file, protein structure file, that is includes the topology information and additional information that you need to provide for your molecular system to be able to do MD. Then we usually do a little minimization to relax the system, to remove high force and hot regions of the system, because you're taking the protein from a natural source. Let's assume that the crystal structure is a kind of natural environment for the protein. So you're taking that from that force field and put it from God's force field and putting it in your force field. So you might not like it, you might have a high energy state, and then that's sort of going to destabilize your simulation. So usually we do some minimization, bring it to cool it down, and then to bring it to a very close, sort of thermally inactive kind of form, and then you start heating it up to the temperature that you want to simulate. Sometimes you have issues here, then you have to go back, do more minimization, or more slowly ramp up the temperature. So in the beginning of the simulation, usually the first picosecond or so, things can get tricky. But be patient, just do some tricks. Maybe you can even take smaller time steps to pass this sort of critical phase, and then after that, usually things are easy. Equilibration. So then you have to equilibrate the system to reach a state in principle. So then that sort of takes some time, depending on the phenomenon, depending on the size of the system, depending on what you want to study. And then after that, then you just, once you have reached equilibrium, you can do your production run and using different ensembles, again, depending on what you want to simulate, constant temperature or constant energy, constant volume or constant pressure. So what is the ensemble you want to simulate? Because we have all these available. So you run that, preferably for under equilibrium conditions, and then you collect the data. That's something nobody can teach you. You have to learn how to make discoveries about your system by collecting the data and analyzing it properly. There are tons of information in the position and velocities of those hundreds of thousands of atoms that you have simulated. It's what you're going to do with them. We show you some examples here, but this depends on your initial question and how hard you analyze the system and try to dig into it. So that's something that is kind of almost on the kind of artistic part of our business. But again, so usually we try to connect the microscopic behavior to something that is measured experimentally, some macroscopic observables, usually average behavior of the system. Let's say if you want to look at it. And under microscopic conditions, things fluctuate much more, of course, you know. But what you're measuring macroscopically, for example, temperature or pressure, is the average behavior, and that's what you want to do with your data. Sometimes there are single molecule experiments that relate very, very nicely and directly to what we do in NAMD. They too are looking at single kind of snapshots of the molecular system. So that would be really nice. They have a distribution. We can compare them nicely to our own distribution coming from the distribution. So I'm not going to repeat this. For initial preparation, QuickMD is a great tool, and you will hear more about that. And again, so I think I would like to emphasize that so many experimental methodologies in structural biology and spectroscopy heavily rely on computing magic. So these are some examples: NMR, X-ray. Just to build the initial model, I mean, these guys also do some sort of simulation or minimization of the structure using similar approaches that we have. So this is one example, actually, in which sort of MD-based methodology was used to prepare capsid structure of HIV virus. Okay, so let me actually now, so I show you some examples of how we use simulations to sort of make discoveries about the mechanism of this. But the first step is to get these structures. So these structures used to come primarily from X-ray crystallography, but then cryo-electron microscopy came along. For a long time, they couldn't do very high-resolution structures, but since a few years ago, they became better and better detectors, direct electron detectors, allowed them to take like 10 to 100 times more samples of the system. So now we have densities, electron microscopy densities that have 3 to 4 angstrom resolutions. So this is really now sort of mainstream source for new structures in the field. So now, of course, I mean, now you would like to be able to use those fuzzy, still fuzzy 3 to 4 angstrom with something like this. You want to actually translate this into a detailed molecular structure with position of all the residues, side chains, and everything. So that's non-trivial. There's a lot of work in the field sort of providing tools that allow experimentalists to do that. One of the applications of MD is in the method of molecular dynamics flexible fitting, something that kind of started here in this center by Klaus Schulten, the former director, and some students, in which they are using MD. As you can see clearly from this figure, they are running a simulation, allowing the molecular system to sort of have its temperature and thermal fluctuation. And then on top of that, somehow, they convert this density into an attractive potential that pulls the molecule inside the density. So that's the idea. Of course, you have to work with it, come up with a nice protocol of doing that. If you do it too harsh, you're going to break things down. So it took us a while to sort of have the optimal way of doing that. But that's actually an example in which you can use MD simulation. So now to, for example, fit structures into density. So I'm using now, this is the original example that we have been using for a long time. And this is a system that Till has been working on, right? This is also from, this is the protozone, right? Yes. Okay. So yeah, so we have some examples that you can see actually that this methodology is now applied to build the structures. So that's one example of how you use MD in conjunction with experimental data. That's what experimentalists want to do. Even if you have no interest in computational modeling and simulating the system, this becomes an extremely powerful tool for you to use. So another example, again, just to motivate you guys a little bit, is when you want to build very, very complex structures, and that's something that the field is going to. So we are moving into cell-scale structural biology. People are looking at the structure of a cell, position of ribosomes, position of mitochondria, the nucleus, and how viruses go inside the cell, how they bud off, and everything. So the field is moving into, at high resolutions, I should say. Of course, we have been able to see how a virus might look like just outside the cell. But we want to have a high-resolution picture of how maybe they get inside the cell, what happens to the cell, what kind of conformational change or configurational change happens in the cell. The whole field is moving into sort of cell scale. And we want to parallel that using computational tools, which means we should be able to put these things together. So these are some two examples that we have. One is chromatophore, a bioenergetic membrane, an organelle inside the cell that has a variety of proteins converting light into chemical energy and then using chemical energy to generate ATP. So that's one example of a sort of organelle which is extremely important. And that's, as I pointed out, this is a model that we would like to prepare using sort of computational methodology. Extremely challenging, both cases. This one took more than 2 years to put together just to have the right packing and a stable system. The same here, but now we have the tools that are sort of semi-automated, allowing you to do these things in a couple of weeks. So this is, let me walk you through this, and the point is not for you to remember all these details. So, but again, so we perform, let's say, for our spherical case, we pack it using Fibonacci algorithm. we pack the lipids nicely here, so using sort of a library of lipids that are coming from an equilibrated membrane. So this, then we take them and place them here nicely. Then we want to distribute our proteins. We have many, many of them, hundreds of them actually in real examples. It's a little bit difficult to work, impossible almost, to do the entire system. So we're going to coarse grain them based on some clustering algorithm. So now I see my whole protein is represented by 10 particles. Some of them are membrane particles. The pink one in the middle, they are supposed to go to the middle of the membrane. The blue guys are supposed to stay in water. I have ways of controlling their behavior by applying different biasing potentials to them. So now I can have multiple copies of these coarse-grained proteins. Insists actually, so they go and partition into my spherical membrane. Those guys are also on the surface, but from this perspective, you might think they're in the middle, they're not. So I can distribute my proteins, again, based on a simple MD, applied to a very coarse-grained system. And they are attracted to these two potentials that I defined. The blue guys are attracted there, and they nicely actually partition your protein there. Then I can take those, put back the real protein, look at the density generated by the real protein, and then try to remove the lipids that are interfering with that density. Either I completely remove them, or I use MD to push them out of those placeholders for my protein. So once you're ready, then you can actually put back your protein in. This is a very, I think, I really claim that this is the most careful way of doing it. We even actually count how many lipids we should remove depending on the shape of the protein. More lipids here, because the protein might look like this, you need to remove more lipids on one side compared to the other, and so on. But the point is actually this very sophisticated or challenging modeling project, we can actually use MD in many steps along the line. So this is the final product. You know, it's almost half a micron wide and composed of 1,400 copies of 16 different proteins and several million, few million lipids, and so on and so forth. So without MD, it's impossible actually to put this together. Whatever method you use, if you don't relax it and refine it along the way, at the end you get something that you simulated for 10 nanoseconds, it's going to collapse. So, and this is another example, and I think after this we can have 9 to 10:30, I think, right? 10:20. So this is another example in which we are taking an experimental electron density of endoplasmic reticulum. And again, we use MD-based steps to create actually an atomic model. In fact, actually what you're looking at is a full atomic model, too large, 4 billion atoms. We couldn't even load this in one piece into BMD. If you zoom in now, you can see this is actually a fully atomistic model we are looking at. Different types of lipids, cholesterol, we can see the black guys are cholesterol with the two leaflets, and you can see the diversity of lipids and heterogeneity of the membrane, which is again a realistic feature. We want to simulate biological system as realistic as we can. So this is an example of using MD to put this together. Again, I repeat, without MD-based approaches or MD refinements done along the way, it's impossible. So this example just walks you through very briefly through what we do. Then we start with the density. So we sort of have to break it down into triangles and use the normals to all those triangles to find actually the normal to the vertex, and then use those to essentially what we are doing here to prepare the inner leaflet with respect to the outer leaflet. And we're going to use the same to place the lipids from a library of yeah, initially actually we represent lipids in a coarse grain manner, do some MD to relax them, you can see, and to sort of evenly distribute them. And then we're going to replace these individual beads or particles with real lipids. We look at their surrounding to find their plane, where they are sitting in, and to identify in which direction the tails should go. Add the lipid here, but you are adding, let's say, a few million lipids in this system. Of course, they're going to clash with each other initially, so we have a lot of what we call ring piercing and clashes. Again, we use MD and additional tricks to relax these bad conflicts here. You can see locally we relax them and refine them. And when we do this for all these bad locations, then we have a system that is stable and we can simulate it in MD. Okay. Okay, so just to sum it up, so we're going to use, let's go now to the, so this was just biological motivation. What are the kind of things you can do with MD? So now let's get into sort of more hardcore of the business. So essentially what you're doing in MD, you're generating a thermodynamic ensemble. So that sometimes that's all you want to do. So I have measured something about the system. I know the bond distance is this experimentally, or the distance between the two ends is something on the average, I don't know, 5 angstrom. I want to have an ensemble, equilibrium ensemble, and then measure those structural or thermodynamical properties and relate them to experimental observables. So you want to take into account fluctuation, dynamic in interpretation of experimental observables. Sometimes you want to actually go after molecular processes, things that might happen. If they happen under equilibrium and time scales you can afford, you're fine, you just run it and analyze your results. But sometimes, and then you can, of course, also get their free energy, which is really something you can directly relate to experimental measurements, and the real behavior of the system. This is something that you can also get from MD. And then, as I pointed out and showed some example, it helps with molecular modeling of different systems because it allows you to do really effective refinement of the molecular structure. So what is MD? I think this is molecular dynamics 1.0. Everybody knows. So what we do is pretty simple at the core. So you know the position of an atom at time t. We know that its velocity, or we assign the velocity somehow, and then you know where it's going to go at a later time. The same happens to the velocity. So if you have the initial velocity, and specify the time step, you can calculate where the position will be at a later time. The same for velocities. So if you have initial velocity, you should assign this based on the temperature. And you know the acceleration on that particle or that atom. Then you can calculate the velocity at a later time, plug it in here, get the position, and so on and so forth. How am I going to get the acceleration? I need to know the total force acting on the atom or particle divided by its mass. Trivial, that gives me the acceleration. So if I have the force, acceleration, I can go on and do this. So now what is force? Force is a little bit difficult to work with. It's a vector, so usually we express that as the first derivative of the potential energy or Hamiltonian with respect to positions. And that's easier to sort of formulate, to discuss, etc. So if I somehow define a set of rules, potential energy, how things are interacting with each other, how much they repel each other, if they're 5 angstroms apart, what is the bond behavior. Somehow I have to describe my molecular system based on a handful of rules. If it's too complicated, I can't do it. You have to do it quantum mechanically, so there your Hamiltonian is much more complicated. But the idea is the same. So I need to calculate the force, so I need to come up with a set of rules, call this the force field. And then once I have that, then I just go on and on. There are additional tricks to make it more efficient, more accurate, etc. But this is the basic. So what I need to do is to go after this guy by having a potential energy function or landscape described for the system. So now this is what I do. And here is an example of the one-dimensional problem. You start from your PDB. That's what I was saying. Your PDB might be a very high energy state. On your potential, on a real system, it might be actually sitting nicely at the minimum energy. But now you put it in your potential, what you define here. It doesn't like it. That arrangement of atoms is very unstable. So it might be here, which means in this case it's going to feel a huge force in this direction and come down. That's too fast for the algorithm that we're going to look at, and it's going to crash. So now we're going to minimize the system, bring it down here, and then we start giving it temperature to allow it to go a little bit more and just go over all these local barriers and then reach the next kind of low energy state. So in this case, this is actually your global minimum energy. But if you look at the other low energy states that are shown here, they all sort of contribute to your ensemble behavior because they are maybe only a fraction of KT higher compared to this guy. We want to be able to also sample these points. So now, this all becomes difficult because going over larger barriers just takes more time. You're giving limited kinetic energy to the system, so the system is going to go left and right. Low energy barriers can be overcome very easily, but these high energy barriers, the probability of the system to reach here and cross it is going to be exponentially lower and lower as you increase the barrier. So sampling and sampling other states becomes challenging. So let me actually point out something right here. So sometimes you say, oh, I need to reach equilibrium and do my production run. That's what we described in the first slide as one of the steps. So what is the equilibrium state in this case? So if you really, I don't know what you're measuring. If you are measuring something that includes the entire thing here, then you have to reach a state where your system is moving between all these back and forth many, many times. That would be your equilibrium state. Then you can actually look at the average behaviors. But sometimes you can't. You can't do that. In those cases, then you might say, oh, well, I mean, my equilibrium state is sampling along this x-axis. I'm only sampling this particular state. Let's say A, B, C, D. I'm only looking at the open state of my channel. And in there, I run this simulation long enough to get enough sampling of this region, of this basin. And that is, for that particular state, I consider that equilibrium. So you are only describing this state. And of course, if you zoom in to this region, you see something like this in there. It's just going to look like sort of micro details in terms of lots of barriers, side chains moving around, loops moving left and right, etc., etc. So really, the definition of an equilibrium state, so oftentimes people ask, well, when do we reach equilibrium? I don't know. It depends on what you're simulating, what observable you want to look at. If you are looking at open channel, open closed transition between these two states, you have to run your simulation long enough to go back and forth between these two many, many times. Only then can you say, is this a lower energy state or that one, right? Because the occupancy of these states is proportional to the energy levels. By one transition, you don't know. Maybe it went to the higher energy state stochastically. You have no idea to compare them, but you need to have enough transition between these states to be able to say something about the energetic difference. So that's something to keep in mind. But okay, back to energy and force. So let's say I have a bunch of particles or bunch of atoms in this case, bunch of planets, and they are, I know their initial positions. Yeah, it's a molecular dynamics, it should be something molecular. Okay, atoms, they are not connected to each other, and then I know their initial position. I know I am going to assign random velocities to all these particles in a way that these velocities correspond, the sum of these kinetic energies, the average corresponds to the temperature at which I want to simulate the system. So I can do that. I can either model this initial state or get a PDB, bunch of argon atoms. I can put them there, assign velocities at room temperature, and now I want to do my simulation. What are the forces? How are they going to interact with each other and affect each other? So there are one type of force is the non-bonded force that is summarized here. You can have this universal kind of force or interaction, I should say. This is the potential energy here that we call van der Waals. So this is due to the fact that two atoms, when they come close to each other, or they have sort of random polarization in one atom, induces a polarization in the other one. So you have two little dipoles, and they attract each other. So at longer distances, they always attract each other. That comes in this form. This is the potential energy function. The functional form we use to describe that attraction, the negative sign, energy is going down. It's proportional to the distance between the two atoms to the power of -6. But then even if you could bring them too close to each other, of course, they start bumping into each other and they repel each other. So that comes in here. R to the -12, it dies with distance immediately, but in very, very low distances, this becomes the dominant force or dominant interaction. So you have a combination of a strong repulsion at very short distances and attraction for every pair in the system at longer distances. So this applies to all the particles in the system. Van der Waals, and this potential form is called Lennard-Jones type of potential that we use to describe the interaction. So there is this epsilon here that also describes the depth of the interaction here. But anyway, so that's one type. The other type is if these guys also carry electrostatic charges, then we have to deal with that. That's in addition to van der Waals interaction. So we use a Coulomb potential to describe that interaction. Charge on I, charge on J divided by R, a bunch of constants. That would be the energy of the system. Of course, the force is just the first derivative of this expression here. And that's the vector connecting with it. So we have a lot of pair kind of interactions in this potential. So that's pretty simple. You can write your own MD code. You know those 4 steps. You can assign some R-mean values. This is just the radius of the system. Assign a charge to it, and then you can run your MD simulation. It's very simple. So now what makes it complicated is if you go from this to a molecular system, now suddenly you need like 5 more files just to put this thing together, because now you have new types of interactions. Those were non-bonded interactions. Two particles moving freely spaced, and they come close to each other, they repel each other at longer distances, they attract each other very weakly. Now you're going to put covalent bonds between them. That's a completely different set of rules. So this force acting between these two, attraction, repulsion, whatever you want to call it, it's going to be now 2 orders of magnitude stronger. So we need to have different rules for bonded interactions, which means in the first place you have to define what is connected to what. And what are the set of bonded interactions or potential that would behave, that would reproduce the behavior of this molecular system. So this is one of the challenges. So we have to define what is this atom, what kind of atom we have here. It's called the atom type. And then what is it connected? Atom 1 is connected to atom 2. Atom 1 is a nitrogen with a sp3 hybridization. Atom 2 is a carbon with sp2, oxygen, hydrogen. Everything has to be defined, and we need to have actually rules. How this guy is interacting with this. Does it matter if there is another atom there? So it should only work with bonds, or should look at angles, dihedrals, and all sorts of things. So your force field becomes actually essentially a set of rules. Let me skip this slide. So the potential energy function, or your force field, There's a bunch of constants going into this expression here. We already talked about non-bonded interactions down here. So now you have in new elements the bonded interaction. You have to define all the bonds and then say what is connected to what. You have to define them. And then for each bond, I'm going to describe them as a spring. The force constant of the spring needs to be specified and the equilibrium value of the spring. Same for angles. Again, I have to define all the angles. That goes into my PSF, protein structure file, where I have the topology information for the system. And then force constant, equilibrium value, etc., etc. And then for dihedrals, I use a periodic function, the cosine function, because they should be able to freely rotate. You don't want to have a harmonic potential for it. So for a dihedral, for every dihedral, I have 1 and 2 and 3 constants describing the behavior. So we have proper dihedral angles. Sometimes they are not enough to reproduce the geometry of the system. So we go to improper dihedral angles. For example, if you want to keep the center planar or in a pyramid-like shape, then we use this because they are not good enough to give us all the geometrical aspects of the molecular system. And I'm sure that even if you include all of these, there are still some details that might not be reproduced exactly or perfectly, but for our purposes, we are fine. But if there are projects in which you are interested in those behaviors, then you have to introduce additional potential energy terms describing those aspects. All right, so you have your potential energy. So this is a function of all the positions. And then what you do, you calculate the force, the second derivative. That gives you the acceleration on each atom. And that's the first derivative of the potential energy function. What you're doing during the NAMD simulation, essentially, for 3n, n being the number of particles, and 3 goes to x and y and z orientations or directions. So you are just calculating this 3n coupled second order differential equation every time step and propagating time. I already pointed out initial structure coordinates obtained from crystal structure for a protein. Velocities, you are assigning them according from Boltzmann distribution. And then most of the time, this is a very common flavor of dynamics that we do, Langevin dynamics, in which in addition to the force that you assign here, there is actually some term that introduces some sort of friction, reproducing or representing what is happening in the bulk of the system, balanced with a random noise term here. It also allows you actually to nicely do the temperature, constant temperature. So we look at this. Let's look at this a little bit more. So let's talk about this time scale issue, the bottleneck. So these are some, this is your sort of logarithmic scale of the time scale going from 10 to the second. And now if you look at what is happening in biological system, in molecular system, so bond stretching is some fast vibrational degree of freedom. It happens on a 10 to second time step. Well, if you want to go actually rotation of side chains, especially if they are buried, hinge bending of some parts of the protein, you have to start to go to nanosecond time scale. But most interesting phenomena happen on a much longer time scales, right? Allosteric transition, one end of the protein talking to the other end, definitely need millisecond of random motion to be able to see how the two ends are feeling the average behavior of each other on a long time scale. Local denaturation, folding, unfolding of pieces of the protein, or rotation of buried side chains actually might take a long time because they are confined in, they depend on the buried motion of the system. So we would like to go into longer and longer time steps, but they are, of course, as we discussed, we are limited by the time step here. So the reason we have to take this short time step is, again, so you have a, let me see if I have a slide here. So, okay, so you see, you take your velocity, your acceleration from the force. So imagine that actually now, okay, so you take your acceleration, you calculate your velocity, and then this velocity is determined how much you move in your time step, right? Okay, velocity time steps gives you the displacement. So now imagine that, okay, so you have something that moves at a very high frequency, okay, very fast, and now, let's say every 10 femtosecond you have a period. And now, at this moment, the force calculated tells you that this guy has to move in this direction. If you take a long time step, instead of moving in its natural range, this guy might go there, right? Because you're giving it more time. Moving at the same velocity that you calculate at that instance, it's going to move too long. When it reaches their next time step, you're going to calculate the force for the same bond. It's going to be a huge force going back because it has displaced too far unnaturally. And that's going to pull it back very, very strongly, very high force. And then you're giving it a long time step. It might even start flashing or going to the other side even. And that's going to kill your simulation because it's going to get unstable. Things are going to diverge. That's a technical aspect of the issue. We can't do it. But if you even think about this, so you have this motion, periodic motion going up and down. In order to be able to describe this period sort of nicely, seeing the up and down and everything, you need to have a few snapshots. You just take one snapshot, you might see it always the same place if it matches the frequency. If you take two snapshots, you might end up in the same place too, comes to zero, back and forth. So you have to take enough snapshots of that periodic behavior to be able to reproduce that behavior. So that's actually sort of another reason for us not to be able to take longer time steps. Hydrogen vibration, or bonds that include hydrogen, when they vibrate, you have periods of about 10 femtosecond. We know that from vibrational spectroscopy, from IR spectroscopy, we know that. We know the frequency and you can calculate how fast they move. So if you somehow get rid of the hydrogen in your system, then you can actually go to longer time steps. Or if you somehow cancel the motion of hydrogens, say, okay, I know the hydrogens are moving as a 10 femtosecond period, but I'm going to ignore them. I calculate the forces, but I don't apply the forces. I keep them there, or I apply some constraints that prevent the hydrogen from moving. And that solves your problem. So now you go to longer time steps, 2, maybe 3 femtosecond, and you start having problems with now other, a little bit slower, but other fast degrees of freedom of the system. So this is the main problem here. That's why we can't take very long time steps. Sometimes actually the idea of coarse grain simulation is that, okay, so let's forget about all these little details, the hydrogens, even actually all the hydrogens, I'm going to put them together into the heavy atom, or even actually a bunch of heavy atoms, I'm going to put them together in very large beads, so each representing 4 heavy atoms and all the hydrogens connected to them. And by doing that, first of all, you remove the number of particles in the system by about an order of magnitude. So you go from 1,000 atom system to 100, and now you're dealing with much shallower potential energy, not very deep, that gives you very high frequency, very shallow, things are moving slowly. Even if you give them 10, 20, 30 femtosecond time step, they're not going to go too far. They're not going to generate a larger force. So that way you can actually reach much longer time scales in your NAMD simulations. So, yeah, but that's one important aspect of this business. Okay, I think I talked about this. It's just a little bit more detail. We bring it down here and give the temperature KT for the system to sample. But again, the hope is to be able to really sample over all these states. So this is one example, very, very simple example, but you can easily extend this to more relevant phenomena in your molecular system. If you watch the orientation of this tyrosine, which in this case might be in a very active site of the enzyme or very important for its function or whatever. So then you run your simulation and then you analyze the system. This guy has 200 residues, but you somehow are interested in the tyrosine because it's an important amino acid. So let's look at the behavior, how it moves. You watch it maybe in your molecular visualization system and you say, oh, it's after some time, it just suddenly did this, stays there. This. And then somehow you see something. It's very powerful to look at your simulation trajectories because you can see a lot. But then you have to make sure it wasn't just you that thinks this is happening. Let's quantify that. I'm going to write a short script in VMD, or it's actually pre-scripted. I said, calculate, measure this dihedral angle. I define the orientation by defining these 4 atoms, C alpha, beta, gamma, delta, and monitor the dihedral angle formed by this. So if you run your simulation only 200 nanoseconds or 400 nanoseconds, so, oh, my system is in equilibrium. Look, it's just going back and forth, fluctuating around this dihedral angle. So this is happy. I have, in this basin, I have sampled enough. You can even calculate the free energy between 100 and minus 110 and 20 and 30 because you have enough sampling going back and forth there. But then if your experimental measurement is done for this kind of time period, maybe you can even relate there. But then if you run it longer, suddenly it goes there. So now you have a different state, different microstate, you could say. So in this basin, I have enough sampling there. You can run it longer and see if you can actually describe this second kind of local minimum. And then maybe if you run it even longer and zoom out, maybe hopefully at some point this comes back to this state. Spend some time here going back up to the other dihedral. So if you have enough back and forth now between these 2 states, then you can start talking about their population and translate that into the free energy between these 2 states. So that's the most natural way of calculating. So but that's a good example of sort of making the points that your equilibrium state really depends on the type of phenomenon that you are trying to describe. In some cases, I have seen sometimes, unfortunately, papers that say, okay, so I'm going to, they have done this simulation, exactly something like this, goes from one state to the other, and now they're going to calculate, oh, look, I did 100, this is 1 nanosecond simulation. I did 1,000 nanosecond simulation, imagine a different kind of phenomenon. Now, if I look at the population of my system, 80%, 85% of my simulation, I was in state A, 15% of it I was in state B. Therefore, this is the population distributions, and the free energy difference between these 2 states is X. But if you think about it, so what if you make this 1,000, 2,000, and this guy stays there for longer and longer times sort of time? So that is going to affect your population distribution. Maybe this is actually a lower energy state. In this population distribution, it looks like a higher energy because it takes a sort of smaller population, but then if you run it longer, maybe this was a bad initial conformation. It just took a while to click out of that higher energy state and reach this lower energy state, and now it's going to stay there for many, many nanoseconds. So you need to really have back and forth transition between these states to be able to say something about their energy difference. That's very important. So this is a very good stretch. If I just zoom in, I can say really good, sort of accurate information about the energy difference within this basin. But then beyond that, I don't know. That's So yeah, so identifying the phenomena that might happen. Two things I try to emphasize here. One is you have to quantify it. And then one is then it helps a lot if you just show people what is it that you're looking at. Especially for a protein, it's a little complicated. So you say, with this distance, so I have no idea what this distance is. If you show me where this distance is sitting, a combination of a molecular image showing what it means for biochemists, biologists, and quantitative data proving that it's not just your eyes that saw or thought saw something. It's real. This is the data. This is the average behavior, etc., etc. This is a very powerful combination. Yeah, so we looked at this. So I'm gonna skip this. Sometimes you have to solvate the system either in membrane or in water or both. So that's some additional step. We have tutorials for this as well in case you need to do a membrane protein simulation. You can actually look at this tutorial. So one step that I pointed out was the assignment of velocities. So positions come from your model or from your PDB, but for velocities, so we use this distribution. So for any given temperature here, so we have this relationship. So you can actually have the probability of the velocities at a particular temperature. So you have this Maxwell distribution of atomic velocities. And you know in X, Y, Z, this distribution corresponds to a particular temperature that we assign the system. In fact, we can use the velocities of the system to calculate the temperature, instantaneous temperature of the system. And that's how it is done. In NAMD and other programs, they just use the velocities to report in the log file what the temperature of the system is. So you can scale these down. You can play with these velocities if necessary. So this is a good way of randomizing your system. Let's say you want to, something that is important is to repeat the simulations, to have multiple trajectories, to have better sampling of the system, and just to make sure that you are not doing exactly the same simulation again and again and again. One effective way of randomizing your system is just to randomize velocities using a different seed number initially. So that happens. You run one simulation where we have one system, you have prepared it, minimized it, it's ready to go. Now you can actually randomize initial velocities using different seed numbers to make sure that they sort of they can be claimed as different simulations. Otherwise, you might be doing the same thing, even though you think you have different simulations. The more effective way would be actually to maybe run this simulation for a nanosecond or for a period of time and then take snapshots from that initial kind of ensemble and see new simulations from this. That would be actually even more effective. But anyway, so that's how we do the temperature. So, and I think ubiquitin is the example that you have in your tutorial. And so, and again, this is now we are starting talking about how am I going to analyze the system. What are things that I should look at when I want to analyze the system? So we have things like RMSD, root mean square deviation. This is very easy. It is supposed to capture everything, which is a good advantage. So you look at everything because this is you look at the displacements of the system, square those, get the mean, and then square root that. So that's going to give you, on the average, how much things move. And of course, you can specify what are the atoms you want to look at. Are you going to look at the backbone only? Are you going to include all the heavy atoms or all the atoms? And we have different meanings. So the great power of this is that it looks at everything. And the great disadvantage of this is that it looks at everything. So you say, oh, things are moving. Yes, but what is it that is moving? So that's a good way of initially looking at the system, showing that, okay, my system seems to be stable. It seems that nothing crazy is happening. The protein is not falling apart. But then if you see something happens here, for example, great RMSD change or going up and down, then they have to dig in deeper and find out what that is. You can play with different flavors of the atom selection or atoms included in that to gain information. Let's say, for example, you have a protein that has two domains, or you have a heteromeric or dimeric system like this. Maybe individual proteins are super stable. If you look at them individually, nothing is happening to them, but they might be moving with respect to each other. So you do RMSD for the entire thing, and you see there is an RMSD increase, but then you do it individually for A, RMSD very low, for B, RMSD very low, but for A, B is very high. That already suggests that they are moving with respect to each other or twisting or whatever. So you can play with this atom selection to actually make more meaningful conclusions about the system. There are two steps here. One is actually superposition. They might be moving the space, so you have to remove the overall translational and rotational degrees of freedom before you compare the positions and calculate the RMSDs, because those are not reflecting internal conformational changes of the system. So you have to use one group of atoms to superimpose the frames, and then you can use either the same group of atoms or another group of atoms to calculate the displacements, internal displacement of the domain of the protein. You can, for example, you can run this RMSD initially. You can calculate this for every individual amino acid in the system, how far things are moving. Maybe there is a loop that moves very much, but then the core of the protein is not moving a lot. You can even actually color map it onto your structure using VMD, using an example that you have in your tutorial. And then maybe the initial thing tells you, okay, this is the most stable part of the protein. Look at the blue guys. This is the core of the protein that moves least. For all the other RMSDs that I'm going to do later, I'm going to use that as the reference. I'm going to only use this part to superimpose the frames, and then I can see better how much the other guys are moving. So you can do kind of districts. Yeah, but that's a very global type. That's what I just said. Then if you do this RMSD per residue, you can actually get information about the system. Oh, look, I mean, this part is extremely stable. I'm looking at the tetramer in this case shown in 4 different colors. There is a stretch of 55 to 65 that shows high RMSD in multiple residues or multiple domains. The ends of the protein are not very stable. That's natural. That's expected. And then in the middle, you have something that is moving more. Even without knowing the structure, this is telling me stable part of the protein, something that is moving a lot, and then again, another stable part, and then this. So you go back to the structure and see, well, yeah, I mean, the ends are both down here, this one end, two ends, and then there is this loop up there that is this guy over here. So you can get some information, put it next to the figure, and that way you have a nice combination of data and what it means for people who might be interested in the structure. So I think this is the time for our coffee break, right? So, okay, so I usually say I should have asked 10 questions before we can go for coffee break. But because it was more introductory material, I'm going to give you a discount. Let's go to 5 questions. Any questions up to this point? Application of MD? Yes.
At this point you just made, how do you know when you've added too many constraints to the system to where, you know, how you talked about your protein's doing this, but your constraint maybe stopped that from happening and you didn't catch that motion?
Well, I mean, now in this case, we are not constraining anything yet. You're right, absolutely. If you want to go to constrained bias simulations, then those become really, really You won't be able, with the degrees of freedom that you're saying, you won't be able to study that, because by definition, you're just suppressing that. So when they say something in the free energy calculation, if you can't sample it, restrain it, if there is a degree of freedom that you can't sample enough, like that dihedral motion, we can't sample it enough, so we are interested in the free energy of the system along a different degree of freedom, let's say. But then this thing sometimes happens, sometimes doesn't happen, and of course these are two different states. This is going to contaminate of calculus. Sometimes it's here, it gives you one number along the orthogonal curve, and sometimes it's here, it's going to affect. If you can't control it, if it doesn't do it equilibrium, if you can't reach equilibrium in that degree of freedom, so we better decide and say, okay, I'm going to fix it at 120 degrees, and then in my paper, all right, I calculate the free energy along opening, closing motion of the protein with this dihedral angle at 120. That's the state I studied. I don't know what happens if it was 150. Or you can do both, 120, and do another free energy calculation for 150 along the same degree of freedom, and then nicely show, oh, look, when it's 120, it has a high barrier to do that. When it's 150, it's much easier to do this transition. So therefore, there is allosteric communication between this tyrus in residue and whatever phenomenon we're studying. So yeah, that's the. Anything else? Yes.
Me? Okay. So in the giant whole cell simulations, you described a very thorough method for placing proteins in the membrane. And whenever I've done membrane simulations, I simply just put the protein in and then delete lipids that are within a certain amount of angstroms from the protein, and then equilibrate that. So is that an improper way of doing it?
No.
Okay.
You're fine. And then this is not a problem if you have a single copy of the protein, flat membrane, things can shrink easily. So very big challenge of this system is that it has an enclosed compartment inside. So let's say you fill it with a number of water molecules. If it's enough, good. If it's not enough, there is no way for water to exchange. I mean, there are ways, but very complicated. So by definition, if you have an enclosed compartment, if your density is low and there is no way for the system can shrink and collapse. This happens. If you don't pack it enough with water, or if you don't have enough lipids in that spherical shape, there is no way that the system can reach a sort of balanced. You see, you have two leaflets, very large, and then in your flat system, the X and Y can freely change. And you have maybe, I don't know, 80, 90, 100 lipids in each leaflet. It's very easy for the system to relax from a bad initial configuration. Whereas for this spherical system, it's going to cause disaster, actually. It's really complicated. And plus, actually, you have maybe 1,000 proteins. If you're off by 2 lipids per protein, that means 2,000 lipids are missing for this whole thing. That translates in a lot of problems. Okay, so anything else at this point? So then after the break, we're going to look at some biased flavors of simulations and what other tricks you can do to get out of this local traps caused by the time scale limitation. So